Developmental biology is the study of the process by which animals and plants grow and develop, and is synonymous with ontogeny. In animals, most development occurs in embryonic life, but it is also found in regeneration, asexual reproduction and metamorphosis, and in the growth and differentiation of stem cells in the adult organism. In plants, development occurs in embryos, during vegetative reproduction, and in the normal outgrowth of roots, shoots and flowers. Practical outcomes from the study of animal developmental biology have included in vitro fertilization, now widely used in fertility treatment, the understanding of risks from substances that can damage the fetus, and the creation of various animal models for human disease which are useful in research. Developmental biology has also helped to generate modern stem cell biology, which promises a number of important practical benefits for human health. Perspectives. The main processes involved in the embryonic development of animals are regional specification, morphogenesis, cell differentiation, growth, and the overall control of timing. Regional specification refers to the processes that create spatial pattern in a ball or sheet of initially similar cells. This generally involves the action of cytoplasmic determinants, located within parts of the fertilized egg, and of inductive signals emitted from signaling centers in the embryo. The early stages of regional specification do not generate functional differentiated cells, but cell populations committed to develop to a specific region or part of the organism. These are defined by the expression of specific combinations of transcription factors. Morphogenesis relates to the formation of three-dimensional shape. It mainly involves the orchestrated movements of cell sheets and of individual cells. Morphogenesis is important for creating the three germ layers of the early embryo and for building up complex structures during organ development. Cell differentiation relates specifically to the formation of functional cell types such as nerve, muscle, secretory epithelia, etc. Differentiated cells contain large amounts of specific proteins associated with the cell function. Growth involves both an overall increase in size, and also the differential growth of parts, which contributes to morphogenesis. Growth mostly occurs through cell division, but also through changes of cell size and the deposition of extracellular materials. The control of timing of events and the integration of the various processes with one another is the least well understood area of the subject. It remains unclear whether animal embryos contain a master clock mechanism or not. The development of plants involves similar processes to that of animals. However, plant cells are mostly motile, so morphogenesis is achieved by differential growth, without cell movements. Also, the inductive signals and the genes involved in plant development are different from those that control animal development. Developmental model organisms. Much of developmental biology research in recent decades has focused on the use of a small number of model organisms. It has turned out that there is much conservation of developmental mechanisms across the animal kingdom. In early development, different vertebrate species all use essentially the same inductive signals and the same genes encoding regional identity. Even invertebrates use a similar repertoire of signals and genes, although the body parts formed are significantly different. Model organisms each have some particular experimental advantages which have enabled them to become popular among researchers. In one sense they are models for the whole animal kingdom, and in another sense they are models for human development, which is difficult to study directly for both ethical and practical reasons. Model organisms have been most useful for elucidating the broad nature of developmental mechanisms. The more detail is sought, the more they differ from each other and from humans. Vertebrates. Frog. Xenopus. Good embryo supply, especially suitable for microsurgery. Zebrafish. Danio ririo. Good embryo supply, well-developed genetics. Chicken. Gallus gallus. Early stages similar to mammal, but microsurgery easier. Low cost. Mouse. Mus musculus. a mammal with well-developed genetics. Invertebrates, fruit fly, Drosophila melanogaster, good embryo supply, well-developed genetics. Nematode, Canorhabditis elegans, good embryo supply, well-developed genetics. Low cost. Also popular for some purposes have been sea urchins and ascidians. For studies of regeneration, urodal amphibians such as the axolotl Ambystoma mexicanum are used, and also planarian worms such as Schmidti mediterranea. Plant development has focused on the Thalicrus Arabidopsis thaliana as a model organism. Developmental processes. Embryonic development of animals. The sperm and egg fuse in the process of fertilization to form a fertilized egg, or zygote. This undergoes a period of divisions to form a ball or sheet of similar cells called a blastula or blastoderm. These cell divisions are usually rapid with no growth, so the daughter cells are half the size of the mother cell and the whole embryo stays about the same size. They are called cleavage divisions. Morphogenetic movements convert the cell mass into a three-layered structure consisting of multicellular sheets called ectoderm, mesoderm and endoderm, which are known as germ layers. This is the process of gastrulation. During cleavage and gastrulation, the first regional specification events occur. In addition to the formation of the three germ layers themselves, these often generate extraembryonic structures, such as the mammalian placenta, needed for support and nutrition of the embryo, and also establish differences of commitment along the anteroposterior axis. Regional specification is initiated by the presence of cytoplasmic determinants in one part of the zygote. The cells that contain the determinant become a signaling center and emit an inducing factor. Because the inducing factor is produced in one place, diffuses away, and decays, it forms a concentration gradient. High near the source cells and low further away. The remaining cells of the embryo, which do not contain the determinant, are competent to respond to different concentrations by upregulating specific developmental control genes. This results in a series of zones becoming set up, arranged at progressively greater distance from the signaling center. In each zone, a different combination of developmental control genes is upregulated. These genes encode transcription factors which upregulate new combinations of gene activity in each region, among other functions. These transcription factors control expression of genes conferring specific adhesive and motility properties on the cells in which they are active. Because of these different morphogenetic properties, the cells of each germ layer move to form sheets such that the ectoderm ends up on the outside, mesoderm in the middle, and endoderm on the inside. Morphogenetic movements not only change the shape and structure of the embryo, but by bringing cell sheets into new spatial relationships, they also make possible new phases of signaling and response between them. Growth in embryos is mostly autonomous. For each territory of cells, the growth rate is controlled by the combination of genes that are active. Free-living embryos do not grow in mass as they have no external food supply, but embryos fed by a placenta or extraembryonic yolk supply can grow very fast, and changes to relative growth rate between parts in these organisms help to produce the final overall anatomy. The whole process needs to be coordinated in time, and how this is controlled is not understood. There may be a master clock able to communicate with all parts of the embryo that controls the course of events, or timing may depend simply on local causal sequences of events. Cell differentiation. Cell differentiation is the process whereby different functional cell types arise in development. For example, neurons, muscle fibers and hepatocytes are well-known types of differentiated cell. Differentiated cells usually produce large amounts of a few proteins that are required for their specific function, and this gives them the characteristic appearance that enables them to be recognized under the light microscope. The genes encoding these proteins are highly active. Typically, their chromatin structure is very open, allowing access for the transcription enzymes. and specific transcription factors bind to regulatory sequences in the DNA in order to activate gene expression. For example, neurode is a key transcription factor for neuronal differentiation, myogenin for muscle differentiation, and HNF4 for hepatocyte differentiation. Cell differentiation is usually the final stage of development, preceded by several states of commitment which are not visibly differentiated. A single tissue, formed from a single type of progenitor cell or stem cell, often consists of several differentiated cell types. Control of their formation involves a process of lateral inhibition, based on the properties of the Notch delta signaling system. For example, in the neural plate of the embryo, this system operates to generate a population of neuronal precursor cells in which neurode is highly expressed. Regeneration indicates the ability to regrow a missing part. This is very prevalent amongst plants, which show continuous growth, and also among colonial animals such as hydroids and ascidians. But most interest by developmental biologists has been shown in the regeneration of parts in free-living animals. In particular, 4 models have been the subject of much investigation. 2 of these have the ability to regenerate whole bodies. Hydra, which can regenerate any part of the polyp from a small fragment, and planarian worms, which can usually regenerate both heads and tails. Both of these examples have continuous cell turnover fed by stem cells and, at least in planaria, at least some of the stem cells have been shown to be pluripotent. The other 2 models show only distal regeneration of appendages. These are the insect appendages, usually the legs of hemimetabolous insects such as the cricket, and the limbs of urodal amphibians. Considerable information is now available about amphibian limb regeneration, and it is known that each cell type regenerates itself, except for connective tissues where there is considerable interconversion between cartilage, dermis and tendons. In terms of the pattern of structures, this is controlled by a reactivation of signals active in the embryo. There is still debate about the old question of whether regeneration is a pristine, or an adaptive property. If the former is the case, with improved knowledge, we might expect to be able to improve regenerative ability in humans. If the latter, then each instance of regeneration is presumed to have arisen by natural selection in circumstances particular to the species, so no general rules would be expected. Metamorphosis. Developmental processes are very evident during the process of metamorphosis. This occurs in various types of animal. Well known are the examples of the frog, which usually hatches as a tadpole and metamorphoses to an adult frog, and certain insects which hatch as a larva and then become remodeled to the adult form during a pupal stage. All the developmental processes listed above occur during metamorphosis. Examples that have been especially well studied include tail loss and other changes in the tadpole of the frog Xenopus, and the biology of the imaginal discs, which generate the adult body parts of the fly Drosophila melanogaster.